Hello, good day, everyone. Thanks for joining us. I can see so many of us here. Ibikunle, the very first person that joined, you're welcome. I can see Olivia, you're welcome. Tolu Animi, Susan, everyone, Jude Eze, you're welcome. Thank you guys for joining us today for this momentous event. As we are moving towards the future of work, it's a pleasure having you. My name is Sophia Jack Dechuku, and I'll be your host for this event. In May 2021, we launched the Hamoye Career Coaching webinar series, and we have since then nailed some very indispensable concepts, including becoming a data scientist, data science applications in cybersecurity, business modeling, completing machine learning projects, amongst others. And we've also brought in data science and machine learning professionals as resource persons to these events. Today is not different. As a matter of fact, today's focus has also evolved around the long-standing debates about the applications of artificial intelligence, AI. The field of AI keeps growing steadily, but today's focus is in medicine. Since 1950, when AI was first mentioned, there have been several limitations in early models that prevented widespread acceptance and application to medicine. In the early 2000s, many of these limitations were overcome by the advent of deep learning. Now that AI systems are capable of analyzing complex algorithms and self-learning, you would agree with me that this session is necessary to enlighten us about its role in biomedicine. Our topic today is artificial intelligence in biomedicine. And today, our resource person is Tobiloba Adejumo, an exceptional machine learning and artificial intelligence pundit who has had an immense impact in the formation of over 2,500 interns in the Hamoye Data Science Internship of 2020 as last year as head of programs. He is currently a doctoral candidate in the field of biomedical engineering at the University of Illinois, Chicago. Before we move on, Toby, we are thrilled to connect with you today and to feed from the depth of your research dexterity. I'm particularly honored to have been led by you. And maybe I should ask this. What was your motivation for biomedical engineering?
Thank you so much, Sophia. So I opted in for a career in biomedical engineering because I felt the need to, solve blindness by early diagnosis.
Okay, wow, that's interesting. Very, very interesting. I'm sure we've all heard that. And I believe we are all itching to hear much more that will inspire us in our career. So before we move on, let's take note of the following. The webinar will last for 1 hour. And as you have noticed, you've all been muted. If you have any questions, please use the Slido event code 6700 on slido.com, 6700. Alternatively, you can post your questions in the chat boxes on Zoom and on YouTube, and we'll be taking all your questions. Thank you, guys. I'll hand over to Toby now.
Thank you so much, Sophia. So let me share my screen. Can you see my screen?
Yes, we can. Yes, we can.
All right. Thank you so much, Emmanuel. So I'm so happy to be here again. Thank you so much, Sophia. Today, I will be talking about artificial intelligence in biomedicine. When I first learned that I'll be speaking about artificial intelligence in biomedicine, I was excited. And after a while, I realized I had spent the past 1 year learning about biology, about physiology, about how cells interact. I've spent the last 1 year having domain knowledge in biomedicine. And then I realized the never ending and never slowing down advancement in AI. With this, I became nervous. In today's discussion, we will be looking at a case study in in which AI is applied in biomedicine. First, I will briefly introduce the domain knowledge and problems associated with the domain knowledge. Then I will introduce the rationale behind the technique that was used for data collection. And lastly, we will examine the results obtained from the experiments. And yes, I forgot to mention my research interests, which cuts across the use of imaging technologies and deep learning algorithms to improve the diagnosis and treatment of retinal diseases. And I'm currently a PhD student in biomedical engineering at the University of Illinois, Chicago. I have a background in computer science, which is why I am also interested in developing biomarkers that can be used in machine learning algorithms for classification tasks in ophthalmology. If you have noticed, I have used both deep learning and machine learning loosely. So I will take a quick detour to define the differences between these two. First, we will look at the difference between data science and artificial intelligence. Then we will examine the difference between AI and machine learning, ML. And lastly, we'll examine the difference between ML and deep learning. Now, data science is a comprehensive process. And when I say comprehensive process, I mean a process that involves pre-processing, analysis, and visualization and prediction. On the other hand, AI is the implementation of a predictive model to forecast future events. We can also say that AI is a broad domain that is still largely unexplored, while data science is a field that makes use of AI to generate predictions, but also focuses on transforming data for analysis and visualization. Another difference that we can see between data science and AI is we build models that use statistical analysis using data science. And on the other hand, AI is used for building models that can emulate cognition and the reasoning of humans. Now, the subtle difference between AI and machine learning is this. With artificial intelligence, we can create intelligent systems, and these systems can mimic or can simulate the human intelligence. In machine learning, we can extract knowledge from data, right? So ML as a subfield of AI, right, enables machine to learn from the past data or experiences without being programmed explicitly. Now, the major difference between ML and data and deep learning is this. While machine learning focuses on enabling computers to perform tasks without being explicitly programmed, deep learning is a type of learning that makes use of artificial neural network. Now, another quick detail to quickly explain what biomedicine means. Biomedicine is a type of medicine based on the application of the principles of natural sciences, especially biology and biochemistry. In modern scientific medicine, biomedicine simply means explaining health in terms of biology. So it is very, very important to really understand the anatomy, which is the body structure, and systems, which is the physiology, in order to understand mechanisms such as the heart, the arteries, the nerves, the brain, and so on. I know that not everyone might be interested in this knowledge, but these things are very vital when we are building a model. I have a quiz in the next slide. So what is the function of an artery? If you know, you can drop your answers in the comment section on Zoom or on YouTube. So what is the function of an artery? Is it A, transports deoxygenated blood, or B, or C, or D? Let me see what, so Suzanne says D, transports oxygenated blood away from the heart. I'm getting A and D, I'm getting C, I'm getting D. Okay, so you can type your answer in the comment section if you're confident enough. You have like 5 seconds. Is it B, transports? Let's see if it's B. No, it's not B. Let's see. Then what about A? Does it transport the oxygenated blood to the heart? No, okay, someone chose A. Let's see if it's A. It's not A. So it's between C and D now, right? So what's the answer? Let's see. So it's D. So artery transports oxygenated blood away from the heart. So let's see the function of the thing. So what is the function of a vein? So now you've had expo, you can easily know, now you can say that D is not the answer, right? Because that's for artery. So what is the function of a vein? Let me see the answers that we have in the comment section. Wow, I've gotten 2 A's. This is amazing. All right, so let's start from D this time. I doubt no one will choose D. So I've not seen any D. I've gotten B. That's nice. From Susan, David says A. Ramon says A. You know, it's nice to get like different answers like this. I've gotten B. Okay, so let me start, revealing the answers little by little. So let's start from D. Is it D? No, it's not D. So let's see. What's the next answer? Is it B? It's not also B. So the answer is between A and C this time. So let's see. Yes, A is the answer. Now, the reason for this quiz is this. Remember, I mentioned earlier that we are focusing on the eye, right? And what we are trying to do is that we are trying to prevent blindness. And as data scientists or machine learning engineers, we cannot outrightly create some kind of liquid portion that can make visually impaired people see, right? However, what we can do, by playing our part is by enabling the early diagnosis of different retinal diseases before they can cause permanent loss of vision, right? And these different retinal diseases affect the artery and vein differently, so that is why it's good that you know the function of an artery, and it's also good that you know the function of a vein. So this particular slide talks about the dissertation that this whole presentation will be centered on. So Dr. Minaj Alam, who was previously a member of my lab before his dissertation, worked on this with so many collaborators, and many papers were published which stemmed from this research. Now, please listen. Different retinal diseases, like I said earlier, affect the blood vessels in the eye differently. This is the known fact. Everyone knows this. They've written papers about this. And when I say blood vessels, I mean artery and I mean vein. And if we can clearly differentiate the artery and the vein, there is every tendency that we can detect early and also stage the different retinal diseases. And by staging, I mean if it's moderate, if it's severe, if it's mild. So we can actually stage these different retinal diseases from a very early stage. And by automated here, I mean, we've talked about automated initially when I explained deep learning and machine learning approach. So quickly, when you're talking about retinal images, that's about the eye. So let's quickly go over the structure of the eye. So as the title suggests of the dissertation, our primary field of interest is the retina. Now, the retina is the layer at the very back of the eye. So this is the retina here. And it enables us to see. So when light comes through the pupil, right, the images are formed on the retina. We can broadly divide the retina into two parts, the inner retina and the outer retina. The inner retina is primarily supported by 3 vascular layers. So this is the retina here. This small part here that you can see here is these are the different layers of the retina. So we can see the superficial vascular plexus, we can see the intermediate plexus, and we can see the deep vascular plexus. That's for the inner retina. And the outer retina is supported mainly by the choroid vasculature. Looking at the photo on the left, we can see the different areas of interest in the retina, such as the optic disc, that's really shiny stuff at the center here. And we can also see the optic disc, which is our blind spot. And we can see the macular region of the eye. And we can see the macular region is responsible for sharp and color vision. And at the very center of the macula, we have the fovea, which has the highest visual acuity. Now, different retinal diseases may affect the fovea, which can cause vision loss. Therefore, it is of great interest to detect these diseases very early. Now, diabetic retinopathy, also known as DR, is caused by high blood glucose, which can cause damage to the blood vessels and affect the blood flow. There are approximately 4 to 5 million people in the world currently living with diabetes. And of course, by the year 2050, it is expected that 6 to 9 million people will be affected by DR. Now, DR is the major cause of vision loss among working adults. early diabetic retinopathy starts with small red dots, which called microaneurysms here, as you can see. And this can progress to leaking blood vessels, causing thickening of the retina and blurry of vision, or new blood vessel growth that can cause bleeding and definitely lead to long-term blindness. Now, DR can be divided into two stages. We have the non-proliferative DR stage and we have the proliferative DR stage. The non-proliferative DR stage is all marked by aneurysms, hard exudates, hemorrhages, and even in this stage, it can be further divided into mild, moderate, and severe. And in the proliferative stage, this is all marked by the proliferation of abnormal blood vessels. Now, 80% of diabetic patients will eventually develop some kind of retinopathy, which is blindness. Therefore, it is of great interest to find ways to actually detect this kind of disease very early. Now, still on DR retinal disease, that is diabetic retinopathy. Traditionally, to observe the retina, ophthalmologists make use of fundus photograph, which is shown in the leftmost part of my slide. Now, a fundus photograph can enable us to see the blood vessels, right, as well as the areas of vascularization. And by vascularization, I mean the growth of new blood vessels. However, our fundus photograph cannot fully visualize the vasculature in high detail. So looking at this, we can see the blood vessels, but it's not showing in high detail. Now, another imaging modality that can be used is fluorescent angiography. And with the help of a fluorescent dye, we can further enhance our visualization of the blood vessels and then allowing us to see areas of new vascularization. However, for FA involves an invasive procedure. So instead of us to make use of FA, we can make use of OCTA, which is OCT angiography. So using OCT, we can clearly see the vasculature in high details and even some areas of vessel dropouts. Now, this kind of asks a question: what exactly is OCTA? So OCTA comes from optical coherence tomography, OCT. OCT uses lights to image the retina. So when you image the retina, you get the volumetric image of the retina. For example, using OCT, we can take a depth-wise scan through the retina, which we call A scans. And if we take multiple A scans across a certain direction, right, we can produce a B scan. And if we're to image this B scan in a perpendicular direction, that's multiple B scans, then we can get the volumetric data of the retina. And the advantage of using this OCT is that it is non-contact and it is non-invasive, unlike FA, where you have to put a dye in the eye. You don't need any dye in your eye. And this is also real-time imaging coupled with micro resolution. Now, where exactly does this OCT come from? So if we're to take multiple images of a particular location of the eye, right, then we average the different images together. We find the variance rather. Now, the pixel with the highest variance will most likely correspond to blood vessels due to high hemodynamic flow, while the pixels with the lowest variance will most likely correspond to static tissue. So looking at this diagram, you can see that for A, which is the fundus photography, is not so clear, and you can see B, which is OCT. But for OCTA, that's C1, you can easily see the vascular information. B shows the anatomical information, but C1 shows the vascular information, right? Because variance has been taken for each B scans, multiple B scans in one location. When variance is taken, we can easily get the hemodynamic blood flow. We can also segment an OCTA image into the different retinal layers, which is not really possible using a fundus image. So C2 here shows the deep layer, right, of the volumetric image. Now, if we were to take an OCTA image, for instance, this OCTA image, right, and we have to perform some kind of digital image processing, right, the first thing we can do is we can try and binarize the vasculature as shown in B. And then after binarizing the vasculature, we can extract the vessel boundaries and create a parameter known as the vessel perimeter index. We can also skeletonize the blood vessels into a single pixel, right? So if you look at this particular, if you look at this, this is skeletonized. So this is like a single pixel. So it was mineralized here, now it's now skeletonized here. And after doing this, you can see that some blood vessels are very tortuous. That means they have tons like this. And then you can calculate the geodesic distance and the Euclidean distance. So when you find the difference, when you divide the geodetic with the Euclidean, right, you can get the blood vessel tortuosity, right? So these are the ways these are the techniques of getting features, quantitative features in OCTA. Normally, whenever we are using data machine learning algorithms, what we normally use is clinical features, and which is something like hemodynamic blood counts. But this was, as reported in the paper, this was the technique that was used, to derive the quantitative features for OCTA. And another area of focus here is the fovea. So looking at the very center of the fovea, you can see that it's almost a perfect circle, right? So this particular fovea region is known as the fovea avascular zone because there are no blood vessels there, right? And for different retinal diseases, this particular circle becomes distorted based on the different stages. So we can also quantify the area and get a particular feature known as the fovea vascular zone, right? And also you can also measure the complexity of the vessel map, of the overall vessel map. This is the superior, the nasal, the inferior, and the temporal. So, and find these different features in these different regions. Also, they can separate these features based on the retina's eccentricity. So this is the perifovea, the fovea, yeah, this is the fovea here. This is the parafovea, and this is the perifovea. So let's keep going. Now, I've explained to you what the fundus image looks like, right? This is a fundus image here, right? And this paper was reported in the dissertation. So what's the technique that was used here is this. So this particular fundus image, right? A is the fundus image here, and the artery and vein source node were identified, right? And a vessel map was now drawn. So this vessel map was now placed on top of the OCTA image for vessel tracing. Now remember that fundus image shows the artery and vein, right? But it is not clearly differentiated. So this technique was used to obtain the full scale vessel map on the OCTA by finding the vessel map, vessel map on the fundus image and overlaying it with the OCTA image and employing a vessel tracking algorithm. Now, the basic principles of machine learning. For retinal image classification, there are generally two segments. We have the digital image processing and we have the machine learning. Using the pipeline above, the users must first perform digital image processing and extract the features of measurement from the image, right? So we've talked about how you can extract features of measurements. Previously, we discussed the quantitative features in OCTA, such as the blood vessel diameter. All these are features that can be extracted from the OCTA image, right? And for different retinal conditions, these features also vary. So once you've extracted the features and you've compiled them, then these features can be used to train a machine learning segment, right? In the machine learning segment can be used to train a machine learning model. And after training, you can now evaluate the model, its performance, and also get the different disease classification stages. Now, the common metric used in machine learning for classification is accuracy, sensitivity, and specificity. Yes. So the sensitivity refers to the model ability to identify cases with the disease, while specificity refers to the model's ability to identify cases without the disease. And after which you can then now evaluate the model's accuracy. Now, this is another study that was published. So this made use of the different features in OCT and OCTA to create a computer-based diagnostic system. So let me briefly explain what this does also. So this study explored the feasibility of a computer-aided diagnostic system using multimodal approach involving both the OCT and the OCTA feature for classification of non-proliferative diabetic retinopathy. Now, the study extracted three OCT features, right? The curvature, the reflectivity, and the thickness, and also extracted OCTA features. We've discussed OCTA features, the blood vessel diameter, the FAZ, foveal vascular zone, and all of that. And also, it also extracted clinical features such as sex, maybe like glycated hemoglobin and the rest. So all of these features were now used with a random forest classifier for machine learning classification. Now, the first random forest performs binary classification, DR and LD eyes, right? So if it's LD, it stops. If it's DR, then the second random forest classifier now differentiates. Is it mild? Is it moderate? Is it, So the second random forest classifier now tells the degree of the DR that the patient has. So this particular study showed a reported an accuracy of 94.7% using OCTA features only. And when clinical features were added to it and OCTA features were also added to it, the accuracy increased to 98.7%. Now, like I said earlier on, for machine learning, you need to extract features, right? But for deep learning, you just put the image into the, the neural network, and then it does whatever it wants to do. So deep learning-based technologies have also been applied in OCTA for so many processes, for OCTA reconstruction, for the noising of the image, because these images always have noise, for segmentation of the different regions of interest. Now, in principle, a CNN can automatically perform feature extraction, thereby reducing the burden for manual feature engineering, right? And some of these features that the OCTA that deep learning algorithms extract, we can't really say, because we are not the ones that extracted these features by ourselves. Now, to train a CNN for a specific classification task that will require a million of images, it is not possible currently, because this technology is still very new. It's a new imaging modality, so there are not a lot of images available. But we make use of transfer learning, that is, training from previous data sets, such as ImageNet, and we can also make use of regularization techniques, that's an image flipping, and the likes. So this particular study, it was dubbed AVNet, that is an automated AV classification, and multimodal process was used involving OCT and OCTA. So OCT images were, let me see. So the OCT image was put into the neural network, same with corresponding OCTA image, as you can see like this. And after these two images were put into the neural network, the fast OCT actually provides the neural network with the intensity information. You can differentiate artery and vein using the OCT intensity profile. But for OCTA, it reveals the blood flow information. Like I asked initially, what is the function of an artery? What is the function of a vein? They have both different functions, which also means that when you get your OCTA image and you try and get the profile, it has like you can easily tell the difference between an artery and a vein using the blood flow information in OCTA. Now, EVNet comprises of two parts. It has the encoder part and the decoder part. So the encoder part takes in these two OCT image and OCTA image, right? And these are put into this. This is put into the neural network. Now, the encoder is equivalent to CNN classification in CNN, right? Which means that you take an image impulse in, and then you perform feature extraction. The decoder identifies and maps the image features to produce the output image. So after performing feature extraction in the encoder stage, the decoder then identifies that this feature belongs to artery, this feature belongs to vein, and then maps it for total and complete AV classification. So in summary, cross-validation techniques have been commonly employed in OCT machine learning processes. So for instance, the data set is divided into training and testing, and the performance on the model is tested and averaged and also reported. So the issue with one striking problem with this approach is that it is relatively new, as we've discussed, right? And we have like few data sets. Another thing is the low interpretability in deep learning, which means whenever we make use of deep learning techniques, we don't really know what is happening. We don't really know the feature that has been used. And that thing again, another issue or thing that was reported in this presentation was that quantitative OCTA opens up unique opportunity to enable AI classification in retinal diseases. So unlike just using clinical features, because most data set that you find on Cargo are made up of just clinical features, right? And you can't really, get quantitative features without really having knowledge of biomedicine or without really understanding what an athlete does. You must really have a proper understanding of the subject matter. So using this kind of approach of quantitative OCTA, there are different opportunities that's been opened, for AI classification of retinal diseases. And many of these studies also, there is like a big bias, right, in these studies because the data set are often from just a particular location. So if federated learning can be employed, different institutions collaborate, and they bring their trained model, then it is possible that it's possible that bias can be reduced. So that brings me to the end of my presentation.
Wow. Thank you.
If you have any question.
Thank you so much. That was an awesome session, a mind-blowing session. And I'm sure our interns have heard a lot and they've learned a lot and greatly inspired too. So we'll be moving into the question and answer session. I'll be taking the first question from Hamida Adekilekun. He says, I was interested in knowing more about biomedical engineering, how you got to be doing doctoral in that field, what degree you had as an undergraduate, and if doing biomedical engineering for masters will be very lucrative in Nigeria. I have a BSc in medical laboratory science.
Sophia, please, can you repeat the question? I got lost at some point.
All right. He says, I was interested in knowing more about biomedical engineering, how you got to be doing doctoral or PhD in that field, what degree you had as an undergraduate, and if doing biomedical engineering for masters will be very lucrative in Nigeria. He studied BSc in medical laboratory science.
Yeah, like you can. I'll start from the first one. So I switched from not really switch. Biomedical engineering has been like my first love, and I changed from computer science to biomed because I got interested in a friend of mine. She is visually impaired, and when I probed further and asked her for the cause of her impairment, she was like, glaucoma, right? And glaucoma is commonly known as the silent thief of sight. So which means when someone has glaucoma, you won't even know. So that was like the first thing that got me really interested in biomedical engineering, trying to find biomarkers that can let us or allow us to identify glaucoma from a very early stage. Sorry? So how we can identify glaucoma at a very early stage. That was why I got interested in biomedical engineering. So the other question that she asked, you're free to doing a master's in biomedical engineering is very fine because you learn about so many disciplines involved in it, and it also depends on what you choose to specialize in. So for instance, if you are interested in a biomedical imaging aspect of it or neural engineering aspect of it, or you okay, so if you are concerned about the tissue engineering aspect of biomedical engineering or the medical imaging aspect of it, or you're concerned about the neural engineering aspect of it, it's all left to you to decide. But I would say that it's always good to since you are in this data science squad, I would say it's good that you probably specialize in, you learn more about biomedical engineering. So it can help you with your pursue. So you can have like a full focused area in data science and understand what really goes on in depth. I hope I've answered your question.
All right. I hope the question has been answered. Hamida. Okay, I'll take another question. The person says, can cures be developed for blindness using AI rather than just early detection and prevention?
I don't think you can develop cure for AI, for blindness using AI, because what is AI actually doing? It's not curing it. You're not curing it with AI, right? You're not curing with AI. What you can, only those that can cure are maybe those that work in the lab, right? But what you can just do is you can just detect early, right? Diagnose early, stage early. You can't say you want to cure blindness using AI. It's not possible. And like, also, you don't even understand what goes on in the eye, because you're just taking data set and just, applying a bunch of different machine learning algorithms to it. You don't really know what goes on in the eye. So it takes like, you can't cure blindness using AI.
Okay. You mentioned glaucoma as your motivation for going into it. And personally, I know some people suffering from glaucoma. So what do you suggest they do? Is it just for going for regular checkups to be able to detect it early? Are there any signs and symptoms? Because from what I understand, if it's not early detected, it attacks the victim fast. So what would you advise they do? Are there things to take to prevent the disease from degenerating so much, or they just have to go for regular checkups at eye hospitals?
All right, thank you so much for the question. What I would say is this: I know that glaucoma affects the optic nerve edge, right? So that is the region where the blood vessels leave the eye, and sometimes, right? I'm talking from my own perspective, right? So I might be wrong. I'm just giving you like my idea. Sometimes, even if you're going, it's good to go for early checkup, right? But before you might see these signs on the images that are taking, using maybe OCT or using other kind of imaging modality, it might not be so like obvious at the time at the time of infection or at the time you just have the disease. So early checkup can work, right? And I'm sure that there might be other like advancements in research from other labs, what they're working on. But it's important, right, that you go for early checkup. And it's also important that there are like early biomarkers that can easily tell the person, maybe most of all these eye diseases, like 80%, like this DR, when you even start detecting it, right? It has been said that 80% of people that have diabetics will eventually have DR. And that's a very big proportion of people. So even if you detect it, it can be reduced. Like how can I explain this? It's like you might have the disease, but then you might not see any biomarker, and it might have reached a particular point, where you now start seeing biomarkers in the eye. So it's important that you go for early checkup, right? To slow down the to slow down the effect of the disease. But it's also important that we develop early biomarkers for glaucoma, like very early enough to actually prevent blindness.
All right. Thank you very much for that. Another question. This is from Gurab Jain. He says, how do you deal with scarcity of data in the health world, specifically when it comes to newer diseases? How do you deal with scarcity of data?
Okay, so in my presentation, I mentioned ImageNet, right? So we make use of publicly available data sets for image processing. This particular paper, like I also mentioned, right, there is scarcity of data. And I said one way we can combat this by federated learning, which means different institutions come together with their data to get like a big bunch of diversified data. So if we can encourage more federated learning approach, even if you are in another country and you want to perform this kind of experiments, most of the data set that you will get are from people that are in that particular location. So it might not be like a general overview of the whole population that might be affected with the disease. So it is still, federated learning is like, I think it's the best way to go, because if you are in Nigeria, you can use your data in your country with maybe data with in another country, just to test your model to be sure that it is actually, the accuracy is not biased.
All right, thank you. Gurab, I hope that answers your question. This is a question from Ebuka. He says, how can one approach the application of AI in other health-related diseases? How can one approach the application of AI in other health-related diseases?
So let me see. So by other health-related diseases, what exactly do you mean? Because like my primary focus is the eye, right? So I'm very much concerned about, or that's where I choose to, that is my research focus. So if it's about different kind of diseases in the eye, okay, let me give you an example. So for instance, we have the retina, and I mentioned that the retina is let me go back here. So I mentioned that the retina is affected, right? This particular retina here, the retina is where is the back of the eye, where images are focused, that we can see. So this is the different areas of the retina here. So if you want to solve any eye-related problem, right, you have to read about it and see for biomarkers here. So if, for instance, now let's say for glaucoma, right, the ganglion cells are not available. For instance, right, you can only find that through research, through eye experiments, through understanding, okay, bipolar cells. So it takes like, it's not, if for instance, glaucoma, you see that the rods and cones have disappeared. So you can only do that through understanding the different retinal regions. So it takes like, you have to just, it's not just putting in data set into the machine and classifying and all of that. You have to really understand what is going on.
Okay. All right. So in other words, any part of the body that one is interested in, the person has to do more research in that area, more research about the cells and organs that make up those body parts. All right. This is a question that is okay about the Hamoye data science internship. Someone is asking, what's the benefits of the Hamoye data science internship? Well, there are so many benefits. The benefits are numerous. One, it makes you job ready. In fact, that's the most important, because everyone goes to school and to become a professional in their respective fields. The goal is to get job. The goal is to offer a service to humanity. The goal is to make impact. So that's what the internship offers you. It makes you job ready. It exposes you to deep learning in the field, deep career progress. It gives you access to so many other experts, professionals in the field. We have webinars that we conduct per time, and experts, just like Toby is speaking to us today, you get to learn more and more. Your programming skills will be enhanced. Your knowledge base in the field of data science, data engineering, and artificial intelligence will be enhanced. And you also have access to career growth. You'll be exposed to what exactly goes on in the field, because you're going to be doing capstone projects. Yes, real life projects, real life practical projects that relate to the field. So I hope that answers your question. Let me take. Okay, another question from Gilead. He says, how can someone who isn't in graduate school get far in this field of machine learning without domain deep projects like this? How can someone who isn't in graduate school get far in this field of machine learning without domain deep projects like this?
Honestly, I have no idea. I don't know because, like, okay, yeah, by reading papers. So, for instance, if you read papers, at the end of these papers, they always have like data available on request. So you can always read papers, and if you want to get the data that was used for the project, you email. They have the procedure there. So I think if you want to really understand what is happening, you have to read lots of papers. This particular project had like lots of papers, and it's like half a decade work by Dr. Minaj, so, and the collaborators. So you cannot even, it takes in like a lot of effort. What I just said in 20 minutes was not, it wasn't not like that. It took like a lot of effort. It took a lot of time to do. So you can, if you want to really understand what is happening in this field, you have to read like lots of papers. If you need their data, just to practice. I don't know if they give, but you can always ask. You can always send a mail to the journal, or the professor that did the experiment.
Okay, all right, thank you for that. I have another question from Emmanuel Ogunwide on YouTube. Says, hi, Toby. Is the paper on quantitative analysis of retina images publicly available? And also, for someone interested in applying AI to biomedicine, what resources would you recommend for starting out?
Yes, the paper is available on Google Scholar. The full publication is available there. You can go and study it. And for someone starting in biomedicine, like I would say, you need to understand. To be honest, I don't know what to say, but I would say you need to really understand like the biology aspect of it. You can't say you want to maybe do AV classification and not knowing what an actually does, or not like really understanding how these things look like. And like there are many decisions that you need to make. So for instance, if you want to use Fundus camera, you should know like the different imaging modality, the advantages, their disadvantages, their level of resolution that they can see through the retina. So I would say just like keep on with the machine learning, but also have it in mind that, or bear in mind that you need to also have like a domain, any field of machine learning that it used to specialize in. If it's education, if it's medicine, you need to have like that kind of domain knowledge to make decisions, because you can't just make decisions like that without understanding what you're doing.
Okay, thank you for that. Another question from Fab on Zoom. He says, I would like to follow up on your work. Can I get a link to this dissertation? Is it possible to?
Yeah, okay. The link it's on Google Scholar. Like I put the full name here, and the author, Dr. Minaj. Where is it? So it's here. This is the name of the dissertation, right? And it's made up of like lots of papers. You can read through the dissertation, see the papers, and if you have any question, you can contact Dr. Minaj.
Okay. All right, thank you. Ramon Ibrahim Olawale from Zoom is saying, please, can we have other diseases that could be detected or diagnosed through AI? Do you have an idea of other diseases that can be detected through AI?
Okay, sickle cell retinopathy. There is sickle cell retinopathy. Sorry. There is sickle cell retinopathy, right? There are papers that have been published as regards that I know of. There is DR, there is Alzheimer's disease. So like you can always go online and type all these things online, and then you see them. But these are the ones that I know of. And they can also even try detecting other kind of diseases. But I'm just concerned about the eye, like I said. I don't know any other disease again in the body except the eye, because that's what I've been focusing on. since graduate school.
All right, thank you. Let me take the final question based on this domain. It says, what are some of the biggest factors to consider when sourcing data to work with? How do you decide on picking one source over another?
So this data that we used for this paper is not publicly available data sets. They're data sets from patients, from subjects in the lab. So you carry out this kind of testing in the hospitals, with different collaborators, and you get this data set and then you quantify it and see how it looks like. So data set that we use are mostly are the ones around the environment. You can't say you use data sets in another region. That's why I spoke about bias and the way we can try and mitigate bias by federated learning when people come together to perform this task.
All right, thank you. I'll take two more questions, but this is related to the internship course. Usman Oladapo is saying, can a beginner enroll for any of the courses? Sure, Usman, a beginner can enroll. We have different levels which you can enroll for. We have the beginner course, we have the semi-advanced course, and then we have the advanced course. So a beginner can learn. Our beginner course has to do with data analysis with computational thinking. So you can enroll as a beginner and learn. And is there any job opportunities at Hamoye AI Lab after completing the internship? Yes, there's job opportunity. There are job opportunities. This internship was advertised with job opportunities as the end goal. So at the end of the final stage of the internship, those who come out tops will be enrolled in a recruitment process and the best will be chosen. So I wish you all the best. So thank you so much, Toby. Thank you so much. It's been a mind-blowing session with you, and I want to say a big thank you again. And I'm quite confident that our students have had a great time and have learned a lot. One more time, thank you so much, Toby. Announcements. Our interns have been working on different projects over the past 3 weeks and are now ready to make their presentations. So do join us next Saturday, 18th September 2021 at 2 p.m. GMT +1 to listen to their presentations. I'm sure you'll be able to learn one or two things from there too. And also, we have been working on building solutions since our launch because we want to build an army of problem solvers. So do stay tuned to our social media channels at Hamoye HQ because a big announcement is coming on September 27th. So everyone look forward to a big announcement that is coming up soon. So to our students and to everyone that has been part of this webinar, thank you so much for your time. I urge you all to keep learning, keep collaborating, and of course, keep your questions coming. We'll be glad to respond to them. Thank you so much and have a nice weekend. Bye for now.